Welcome to the Sans Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. And hey, if you enjoy what we do, please take a second to like and subscribe. It really does help support the Sans Science Experience. Today, we are diving deep into some really fascinating research that might just shake up how we think about Alzheimer's disease.
Absolutely. We're looking at a study that proposes a pretty surprising link between the energy factories in our cells, mitochondria, and the development of Alzheimer's. And even more intriguing, there's a potential new therapeutic angle emerging from this.
Right, so our mission really is to explore this idea. Could problems with mitochondria actually be a root cause of Alzheimer's, not just a side effect? And we'll also dig into a specific compound that seems to reverse some damage, plus some really critical differences between males and females.
Yeah, it's important context because for decades, the main story in Alzheimer's research has been the amyloid cascade hypothesis. You know, the idea that sticky amyloid beta proteins build up and cause neurons to die.
But the treatments based on that idea, the anti-amyloid therapies, well, they haven't been the home run everyone hoped for, have they?
Not really. They show some effect, sometimes, but often quite modest cognitive benefits. Which, makes you wonder, are we missing something earlier, something more fundamental?
And that's where this new research comes in, pointing squarely at mitochondrial complex 1, or MTCI. It's a key part of how mitochondria make energy. What they found is pretty stark. Just cutting MTCI activity by half seems to be enough to kickstart Alzheimer's-like changes in the brain, molecularly speaking.
Exactly. They demonstrated this using a specific mouse model, mice engineered to have this MTCI deficiency, the NDUFS4 knockout mice. And these mice, they showed all sorts of problems. Mitochondria weren't working right, energy metabolism was off, genes related to synapses weren't being expressed properly.
Sounds a lot like Alzheimer's.
It really does. The gene expression patterns, what scientists call transcriptomic signatures, were eerily similar to what's seen in human late-onset Alzheimer's and other Alzheimer's mouse models. And crucially, like you said, this was happening without the classic amyloid buildup or the tangled tau proteins we usually associate with the disease.
So that really flips the script, doesn't it? It suggests the energy problem might come first.
It definitely puts mitochondria center stage as a potential early driver.
Okay, so if reduced MTCI activity causes these problems, can boosting it or somehow intervening fix things? This is where that compound CP2 comes in, right? But it's an inhibitor.
That seems backward.
It does sound counterintuitive, I agree. You have low activity, so why inhibit it further? But CP2 is a mild inhibitor. And the idea is that this mild stress actually triggers beneficial responses in the cells. Protective pathways get activated.
Like how exercise puts temporary stress on your body but makes you stronger overall.
That's a great analogy. It's a bit like a concept sometimes called mitohormesis. CK2 seemed to, well, it encouraged the cells to make new mitochondria.
Improve how things are transported within the neurons, and importantly, clear out damaged mitochondria, which is a big problem in Alzheimer's. It reduced markers of that damage.
So it's kind of like a cellular quality control boost triggered by a small nudge.
Precisely. It activates these inherent neuroprotective mechanisms.
Now, you mentioned sex differences earlier. That seemed like a really significant part of the findings, too. How did males and females respond differently?
Yeah, this was quite striking. The female mice with the MTCI deficiency, they initially presented with what the researchers described as a sort of milder Alzheimer's-like molecular profile.
Milder, okay.
And importantly, when they were treated with CP2, these female mice showed a much better response.
Yeah.
A more significant reversal of those Alzheimer's-like gene signatures, particularly things related to inflammation and neuron function.
Yeah, but
The male mice, unfortunately, they showed signs consistent with a more severe Alzheimer's profile from the start, just from the MTCI deficiency. And while CP2 did help improve some aspects of their mitochondrial function, it didn't really turn back the clock on the overall gene expression changes as much as it did in the females.
Wow. So the same deficiency, the same treatment, but pretty different outcomes based on sex.
Exactly. It really underscores that biological sex can fundamentally change how the brain responds to energetic stress and potentially to therapies.
So pulling this all together, what's the big takeaway for you listening in?
I think the main point is that mitochondria might be much more than just, innocent bystanders getting damaged in Alzheimer's. This research strongly suggests they could be right at the heart of the disease process, maybe even initiating it. And that makes them a really exciting target for new treatments.
Yeah, a whole new avenue, potentially.
And that leads us to a final thought, something for you to chew on. How could these findings, especially these really stark sex differences in both the disease model and the treatment response, change how we approach Alzheimer's down the line?
Right. Does this mean we need completely different diagnostic tools or maybe different treatments for men and women?
It certainly raises the question. It points towards a future where personalized medicine for Alzheimer's might have to seriously account for sex as a biological variable. What do you think?
Definitely food for thought. It highlights how complex this disease is, but also potentially offers new hope. Well, that's all the time we have for this deep dive. Thanks for joining us on the San Sunnius Experience. Remember to like this video and subscribe to the channel if you found this interesting. It helps us keep bringing you these scientific explorations. We'll see you next time.